So cleaning up the products of cell death is just not gonna cut it. And so you have to fix the refrigerator, right? You gotta get deeper into it. So that's where my idea was, let's start with stem cells in the brain. Now, stem cells have been injected intravenously. They've been injected even into the spinal canal, into the spinal cord area, right? The thecal sac. Um, if you inject something into the vein, it's really not going to get its way up into the brain. Okay, there's the blood-brain barrier. There are the lungs that the venous flow goes to after you inject something intravenously. The cells probably will not propagate much farther than that. They could have a paracrine effect, an effect outside of the cells literally having to get into the brain. They could have some other effect, but um, I wasn't buying it. That's I don't. I'm not buying that. The cells, you inject them into the spinal canal. I don't know whether you know about spinal cell flow. Excuse me, spinal fluid flow, but it flows from the bottom of your spine up and around the outside of your brain, and then gets absorbed into the veins, into the vascular system. So those cells, even if they made it up against gravity, from being injected down here and not just accumulate at the bottom of your spine, they probably aren't going to get in your brain either. So, what people, a lot of people in your audience don't know is the brain is hollow. The brain is a hollow organ. I tell people it's like a honeydew melon. You cut the melon in half and it's hollow in the middle. Our brains are exactly that way. There are hollow areas in the brain called ventricles. And the ventricles produce cerebrospinal fluid. And cerebrospinal fluid is what you get out of a spinal tap. It's the same fluid that your brain is floating in. Our brains are bobbing in our heads right now. They're in our skull, our brains are just bobbing. We're like in a sack of water. And that fluid, believe it or not, is now being proved to literally wash our brains and clean out the debris on a regular basis. Even more so at night while we're sleeping, apparently. We clean our brains even more, and that's why sleep is important. If you could inject the stem cells into that fluid, perhaps the cells will get into the brain more readily, let's say,
Mm.
a little more easily than if you tried IV. But even beyond that, what's cool about the brain, and this is really the most amazing part, is that every one of us, uh, we all have stem cells sitting in our brains right now. And where do they live? Where are these stem cells in our brain? They're around the ventricles, the
Mm.
hollow cavities. And the areas that the stem cells are in are called the subventricular zone. So subventricular, under the ventricle. So if you're inside the ventricle and then you hit the wall of the ventricle and you go through that wall, you're going to be into a zone of a bunch of stem cells. And where do those come from? Those came from embryology. When we were little babies, those stem cells are what formed our brains. And they got up there and then they migrated into the positions they're in. So it's a very fascinating little scene up there. Now, these stem cells are still in us as adult, old human beings. Why don't we use them? What are they doing there? Well, if you look at the research, we've done a lot of research on rats. And, if you're a rat and you can't smell, you're going to die because
Mm.
you're not going to be able to smell your prey and you're not going to be able to smell your food, right? So you're just not going to make it. So rats have evolved a way of replenishing their olfactory neurons, their sense of smell neurons, using their own internal stem cells.
Hmm.
This is well described over the past 10, I'd say 15 years. We've known about this ability of the rat to literally like call on those stem cells. Hey, I need a new sense of smell here. Can you replenish this so I can live longer and survive? So if they can do it, why can't we do it? And the other thing is, why aren't we doing it? I mean, if someone has Alzheimer's disease, why isn't a stem cell fixing the problem?
Mm.
Well, it's my theory that after 60 years, 70 years of age, these stem cells are sitting there. They're tired, they're dormant, they're old. They're not doing much, so they're probably not you. I think we've out evolved our ability to use these. So my theory was, if we injected stem cells into the brain, into the ventricles, and they were able to percolate in there and then turn on those other stem cells, then maybe we could wake them up and get them moving and get them helping to restore a problem. I work with a very famous stem cell expert. His name is Hans Kierstead. He is extremely world famous. He is the father of stem cell technology in America for 30 years. He happens to have a lab about a mile and a half away from here. So he and I have known each other for 20 years. And I said, Hans, does this make any sense to you? He says, yeah, well, we'll use Wnt activated stem cells. And I go, sounds good. What's Wnt?
Right.
So for someone like you, Don, you may know, but I'm just a brain surgeon. I don't know anything about Wnt. And now
No.
I'm like a Wnt expert. So what Wnt is a signaling protein that, if we see it on a stem cell expressing Wnt, that stem cell is turned on and has the ability to turn on other stem cells. Without Wnt expression, the stem cells stay dormant. So if you were to inject Wnt expressing stem cells into the brain, maybe they would percolate into the brain, stimulate the other stem cells to turn each other on, and so forth. That's called the Wnt wave. Kind of like the wave at a football game, where
Right.
people start the wave. That's what happens with the Wnt wave. And some people call it a Wnt cascade. But these cells are that's their job. That's what Wnt's job is to turn on other stem cells and get them awakened and doing something. So what are we doing when we inject stem cells? And I've done work with pigs, I've done work with animals, smaller animals, and I've done work in humans before the FDA trials. And everything is pointed to this being something of interest. So that's why I said, first of all, I have a very nice family. I have a good reputation in my community. I'm well respected here. I have very good, reviews on Google. You don't want to ruin this with snake oil and something crazy. So the only way to legitimize this is to go through the FDA. So we presented our work, we presented our research to the FDA. The FDA said, no, we want you to do the work on pigs first. So I literally went down to a vivarium in San Diego and operated on 14 Yucatan pigs that are, 90 pounds, and put reservoirs in their head. And that's, by the way, how we're injecting the cells. We put a little port under the scalp, and that tube goes into the brain through the skull, into the brain, into that ventricle, into that hollow cavity. And we can tap that port anytime. No one sees it. It's under the skin. It's invisible, but it's very modern. It's very kind of disruptive type of technology, But I mean, if Elon Musk can have little electrodes sticking out of people's heads, we can have a little port underneath the skin.
Right.
And by the way, all of these stem cells are from the patient themselves. These are not off the shelf stem cells. And that, to me, did not make any sense either. You take somebody else's stem cells, you inject them into the brain. I mean, my gosh, it's going to be a graft versus host reaction. You're going to have some sort of allergic reaction to them, and it's just not going to be good. You'll have some inflammatory response. So off the shelf, not good right away. I mean, maybe in the future we can figure that out. How about things like exosomes and other things? We can get there. But I figured the FDA would be okay if we took the patient's own fat from the same patient, grew those cells in culture, selected them for their expression of Wnt, and then used that as the pharmaceutical to inject into the brain.